So the obvious question is what causes sterile intra-amniotic inflammation? And in general, the causes of sterile inflammation are alarmins, senescence of cells, and cell-free DNA. And alarmins are the favorite candidate, and it's important to be familiar with them because we all have alarmins released as we age. So alarmins are released after non-programmed cell death. They can be produced and released by immunocytes without dying. I'll show you that in a moment, and can recruit and activate innate and adaptive immune system. And they restore hemostasis by promoting healing of the tissue. So it's intended to be a homeostatic phenomenon. But as everything in the human body, when we have excessive sterile inflammation caused by alarmins, that causes harm. So here is an inflammatory cell that can be activated by microbial products such as endotoxin or cytokines such as interferon gamma or TNF-alpha or IL-1, and then they induce the production of alarmins, and the prototypic alarmin is HMBG1, and this in turn would activate target cells. Now this is an inflammatory pathway. The non-inflammatory pathway is any cell in our body can undergo apoptotic cell damage or can undergo necrosis, and then during the course of these processes they release alarmins to induce repair. So this is what alarmins are. Now the way this works is, imagine when it says danger signal, that is an alarmin that interacts with a pattern recognition receptor. It goes to the nucleus, it produces a number of proteins, and they include pro-inflammatory cytokines, and the emphasis here is in IL-1 beta, because this is a key cytokine in the inflammatory response. Now, in the title of my talk are inflammasomes. So what is the connection between alarmins and inflammasome? The mechanism responsible for the induction of inflammation in sterile inflammation is activation of the inflammasomes. So what are inflammasomes? Well, these are inflammasomes. Alarmins, when they get into the body, they can lead to assembly of this molecule that is called an inflammasome, release of caspase-1, and caspase-1 cleaves pro-interleukin-1 alpha beta that is not active to active or interleukin-1 beta. So in looking at patients with premature labor and sterile inflammation, we look for the usual suspects, and these are urate crystals, hydroxyapatite crystals, ATP, and a bunch of other things, and all our studies were negative. So we could not attribute premature labor with sterile inflammation to these non-proteinaceous alarmins. But we did find that interleukin-1 alpha and these other alarmins were increased in patients with premature labor and sterile inflammation. So let me tell you two things about HMBG1. It's a prototypic alarmin that is released after necrosis or apoptosis. It's a late mediator of sepsis, often even in the absence of infection, and blockade of HMBG1 reduces the mortality of sepsis. And here you have evidence of an increased concentration of HMBG1 in patients who have premature labor with and without infection, including patients with sterile inflammation. And here are patients who have no inflammation, patients who have infection, because it's proven in red. Here are patients who have sterile intra-amniotic inflammation but low concentrations of HMBG1, and these are the ones who have high concentrations of HMBG1. The point is that they live soon. Now, can alarmin induce premature labor? Here is an experiment in mice in which we did an amniocentesis, injected HMBG1, and 50% of them delivered preterm. What are the mechanisms whereby sterile intra-amniotic inflammation induces premature labor? Here is the inflammasome and how it works. Crystals, but also hypoxia or disturbed flow, activates this complex that is the inflammasome, and then they cleave pro-interleukin-1 alpha beta into active interleukin-1 beta, and we have inflammation. And this is all the papers, and they will be on your syllabus, so if you want to look at them, supporting that the inflammasome has a role in spontaneous labor, but also in sterile inflammation. Can we inhibit the inflammasome and prevent preterm labor? The answer to that is yes. Here we induce premature labor in pregnant mice, giving them an alarmin, and then when we give a specific inhibitor for the NLRP3 inflammasome, we prevent premature labor. But you are an neonatologist, so I imagine that you are wondering, does this have any consequences for the fetus or neonate? So the data that I have is fundamentally in pregnant mice. And what we did is to do a randomized trial in which we gave an alarmin via amniocentesis to pregnant mice. And this is the control group. And the control group, they were all born alive and everything was fine. But this is the group that had sterile intra-amniotic inflammation. All of them were born dead. And if you look carefully, it's not easy to see. These mice, baby mice on the right, are very dry. So they are sick, although we don't know specifically what is. I think that this is systemic inflammation causing non-viability. And here is mortality, suggesting that we can reduce mortality and so on. So can we prevent the mortality by blocking the inflammasome? The answer to that is yes. So here is a therapeutic option in which you can block sterile inflammation by a specific blockade of the inflammasome, and that reduces perinatal mortality. Now, what are the take-home messages? First, we tend to believe that culture-negative sepsis is rare. Is that condition that we identify the babies are sick in the nursery, but the cultures are negative. What is that condition? Well, now we will call that sterile systemic intra-amniotic inflammation if we refer to the fetus, and I think that there is a need to coin that term in the neonate. And those neonates are often born to mothers who have sterile inflammation. They have elevated cytokines in peripheral blood, CRP, they have phoenicitis, but they have no evidence of infection when we look with molecular methods. Can sterile inflammation be treated? I told you before, I showed you evidence that that is the case, but we have evidence from other animal models that we can reverse intra-amniotic inflammation, and I will be glad to discuss how we can do that. I want to remind you that we can treat congenital neuroinflammation with anti-inflammatory agents without giving antibiotics, and that neuroinflammation can exist with infection or with sterile intra-amniotic inflammation. Thank you very much for your attention.